Okay, hello and welcome everybody. Today we're going to be talking about our last set of research design options. These are our quasi-experimental, factorial, and single subject research designs. We'll start with the quasi-experimental research designs, and we'll start with a little bit of a video.
A quasi-experiment is designed a lot like a true experiment, except that in the quasi-experimental design, the participants are not randomly assigned to experimental groups. In a true experiment, research participants have an equal chance of being assigned to any condition of the independent variable, the one being manipulated by the researcher, that is involved in the study. So, for instance, if a researcher was examining the effects of caffeine on reading comprehension, she might randomly assign participants to one of three independent variable conditions: those who drink 1 cup of soda, 2 cups of soda, or no soda. She might then assess each person's reading comprehension abilities following exposure to the independent variable. In a true experiment, each participant who volunteered would have an equal chance of being assigned to any of the three groups. Quasi-experiments are employed when the researcher is interested in independent variables that cannot be randomly assigned. Usually, this happens when the independent variable in question is something that is an innate characteristic of the participants involved. Let's look at a couple of examples to help illustrate the point. Here are two examples of simple quasi-experimental designs that will help you see the difference between a true experiment and a quasi-experiment. Dr. Jones is a personality expert who studies the impact that personality traits have on intelligence. For the purposes of her current research project, she is interested in examining the IQ scores of people who score highly in each of the big five personality factors. Each of the 5 personality factors are a quasi-independent variable. Personality traits are inherent to each person, so random assignment cannot be used. Participants would initially be assigned to groups based on their personality assessment score across each of the 5 personality factors. Now that Dr. Jones has her participant group assignments, she can examine the impact that personality factors may have on intelligence. If a true experimental design were used, each participant would be randomly assigned to each personality group, regardless of whether or not they possess those personality traits, which would not really address the question that Dr. Jones is asking.
So there's a nice little video to get us reoriented to what an experimental versus non-experimental research design is and how that then turns into quasi-experimental. So the key difference in quasi-experimental designs is that they look like experimental methods. They use as much experimental methods as possible, but they lack the ability to randomly assign participants into treatment groups or to option groups. And that key lack of that element of random assignment really changes how much we can take away from that experiment. So, unlike a true experiment that can try to assess causality, quasi-experimental studies cannot conclusively determine causal relationships due to potential confounding variables, and because essentially you can't hold one specific variable different. So in experimental research, we are able to have that random assignment, but in quasi-experimental, we can't have random assignment, which means they can't have causal outcomes, or we can't use statistics that confidently say this, A causes B, because it doesn't have the random assignment piece of it. There are several different types of quasi-experimental designs. So this first one is called non-equivalent group designs. And this is when a research design compares outcomes between two groups that are based on non-random criteria. So this is usually done when there's an ethical reason or a practical issue that prevents random assignment. An example of something like this might be, well, like they talked about in this video with the different personality types and how that's not something you can actually change. So if I'm looking at personality types within people who work at Google, that's my whole sample. I can't pick other people based on their personality type, so I just have to go with what is there. And because of that, I can't randomly assign places because there's a finite amount of each type of participant. That's just one specific example, but the key part of it is non-equivalent group designs can't have this random criteria, so they have to be based on non-random criteria, and that's what prevents the random assignment because you can't. The next type of quasi-experimental design is called interrupted time series. This is when an approach analyzes change in data collected at multiple points in time before and after the intervention or experiment happens. It's valuable for assessing longitudinal changes like policy effects or educational reforms. Remember, longitudinal research is research that studies something over several periods of time, whereas cross-sectional is a snapshot at one time. Interrupted time series is a type of longitudinal research that is quasi-experimental because it can't have that random assignment element of it. But it's still looking at both before the intervention, and after the intervention. This next one is called propensity score matching. This is used to approximate the effects of treatment by matching subjects in treatment and control groups based on covariates that predict receiving the treatment. This method aims to reduce selection bias and approximate the conditions of randomized control trial. Another way to say this is propensity score matching uses a higher level of assessment when deciding what participant should go in which group, but it's not truly random assignment. So if I had 100 people and I wanted to do random assignment, I might just put them in a random order and say 1, 2, 1, 2, 1, 2, 1, 2. And I'm not accounting for variables that I want represented equally. We just randomly assign them. Propensity score matching is when we're going to get a lot of information about those participants and then put them into groups based on that information. So we're still trying to make the participants' key demographics or key experiences even between groups, but we aren't actually randomly assigning them. So it's a more controlled form of assigning. It's supposed to replicate controlled assignment, but it's not true controlled assignment. Or controlled assignment. Quasi-experimental research designs. Again, the real key here is just knowing that it lacks random assignment. Some of the advantages and disadvantages of this. Well, some of the advantages of quasi-experimental research designs is that they're really useful for real-world settings when you can't randomize things. It's just not possible. It's unfeasible. And so it lets researchers study more natural environments, which is really important in behavioral sciences. As you can imagine, it is not always ethical to change people's experiences, especially if it's something harmful. So like, let's say I wanted to study the impact of taking or using heroin once per day versus 2 times per day versus 3 times per day versus none per day. It would be unethical for me to get participants and then start forcing them to use heroin 3 times a day minimum. That's unethical. So using some sort of quasi-experimental where I can't control some of the variables, so I can't truly randomly assign all my participants, but maybe I use this propensity score matching with all my people who are using heroin in my research. And instead of saying random assignment 1, 2, 1, 2, 1, 2, I instead say, okay, well, I have 10 of my 100 people who grew up in Tennessee. I have 10 of them who grew up in Kentucky. So let me put 5 in each of my control groups, 5 in A, 5 in B, so that it's even across these key points of interest, like where they live. So that can be an advantage because in the real world, you very often can't randomly assign people to have different lived experiences. A disadvantage of that is that there is a much higher risk of something like selection bias, and there's just a lot of less control over confronting variables because you aren't able to do that random assignment. And because you can't control it quite the same way in a quasi-experimental, there's just more variables involved. We can't actually confidently say that this one change, this one variable impacted the outcome. We can say it's likely, we can say there's a relationship between them, but we can't say one causes the other. That's an inherent barrier or disadvantage of quasi-experimental research designs. In behavioral sciences, this is used very, very regularly, frequently utilized to evaluate the impact of all sorts of different types of interventions. So societal or social, educational, psychological. It's also true for some medical things. And they're especially prevalent in studies where controlled experiments are impractical, or the testing how effective something isn't ethical to only give to some people and not to others. Some things you need to consider if you're going to use a quasi-experimental research design. There are certain criteria for why you would want to use this. One is ethical constraints. So again, if you can't ethically randomly assign your participants, or you can't ethically control something in that environment, you can't have someone do something for whatever reason that's harmful, this can be a research design that allows for you to still control for what you can, but it's more limited with the randomness, which makes it inherently less able to predict causality. So ethical constraints is the first reason you would think about doing something like this. Logistical limitations, so situations where it's just not like logically possible to do random controls. So it's not just that it's unethical. It might also just be really improbable or not able to do. Could be really expensive to do a true random control study, that sort of thing. And this is often a reason why quasi-experimental stuff is used in large-scale field settings when they're looking at a lot of people all at once, especially in natural environments. That tends to be quasi-experimental because it's just people are really complex, and especially when you need a big sample size, you need a lot of people to participate. It is very hard to do true experimental design because it costs a lot of money and logistically is really challenging. And the third reason you might choose this approach is got to do with the type of question that you're asking. So if your research question can't, or your hypothesis cannot be tested by directly manipulating something, then you might not need to use an experimental design. And so quasi-experimental design might be a better fit. That being said, we know there are these inherent disadvantages, these drawbacks. There are some strategies you can use to try to enhance the validity of a quasi-experimental design. The first one is going to be multiple pre-tests. So collecting data before you even actually do your intervention, before you do the real experiment or measure the real point of interest, test it out several times ahead of time so that you can have a really solid understanding of baseline and make sure that you are reducing variability. Remember a couple of lessons ago, we were talking about how variability is the enemy of science. We want things done the exact same way. The method should be the same. That way we can make sure that everyone gets the same intervention so that we can more confidently say, yeah, it's the intervention. It's not something about the person talking to them, or it's not something about the doctor delivering the intervention. So we want it to be as consistent as possible. Pre-tests are a really great way to be able to practice the intervention and get it down to being very consistent and same. So pre-test we love, especially for quasi-experimental. The next strategy is matching techniques. So this is when we use methods like propensity score matching, talked about last slide, to create comparable groups based on relevant covariates, therefore simulating the conditions of randomized experiment. I used that example a second ago, but if I can't do a true random, it might not be a bad idea to then look at everyone in my participant pool, decide what are the key variables of interest, like what might make these people report something differently, and then making sure that they're about even between my control groups. And we have statistical control abilities as well, different types of statistical techniques that can control for confounding variables or include the understanding of confounding variables. And those types of statistics tend to increase internal validity and external validity as well. The bummer is that it usually requires a much larger sample size, so you need a whole lot more people to participate in the research to be able to do those types of statistics. Next, we'll talk about this factorial research design, and we will start again with a little video.
An important type of experimental research design is the factorial design. In a factorial design, several independent variables, also called factors, are investigated simultaneously. For example, we could investigate the effectiveness of an experimental drug aiming to reduce migraine attacks. Suppose we create three conditions that differ in the administered dosage: low, medium, and high. We can now investigate the effect of the factor dosage on the number of migraine attacks. Is a higher dosage more effective in reducing attacks? We can extend the simple design and make it factorial by adding a second factor, for example, gender. If we make sure there are enough, preferably equal numbers of men and women assigned to each of the dosages, then we end up with 6 conditions. men who receive a low, medium, or high dosage, and women who receive a low, medium, or high dosage. Besides the effect of dosage, we can now also investigate the effect of the second factor, gender. Do women suffer more migraine attacks than men? We can also investigate the combined effect of dosage and gender. We can see whether a higher dosage is more effective in reducing the number of migraine attacks for women as compared to men. The effects of the factors separately are called main effects. The combined effect is called the interaction effect. In this case, we're dealing with a two-way interaction because the effect combines two factors. Of course, we can add more factors, making it possible to investigate higher order interactions. But as the number of factors increases, the design becomes very complicated very quickly. Suppose we add diet as a factor with two conditions, a normal diet and a diet eliminating all chocolate and red wine. Let's call this the no-fun diet. This would require that each of the 6 groups is split in 2, with half of the participants being assigned to the normal diet and half to the no-fun diet. We can now look at the main effect of diet. Is a no-fun diet effective at reducing migraine attacks? We can also look at the two-way interaction between diet and gender. Maybe the no-fun diet is effective for men, but not for women. We can also look at the two-way interaction between diet and dosage. Is a higher dosage more effective when a no-fun diet is being followed as compared to a normal diet? Finally, we can look at the three-way interaction. Maybe a higher dosage is effective for women, regardless of diet, but maybe for men, a higher dosage is effective only if they follow a no-fun diet, and not if they follow a normal diet. Like I said, it can get pretty complicated pretty quickly. There are even more complicated factorial designs called incomplete designs, where not all combinations of levels of the factors or cells are actually present in the design. Now, we won't go into those designs right now. What's important here is that you understand the basics of factorial designs. You need to know that a factorial design consists of 2 or more independent variables and, for now, 1 dependent variable. The independent variables are crossed to ensure that all cells are represented in the study and that each cell contains enough participants. Factorial designs are very useful because they allow us to investigate not only the main effects of several factors, but also the combined effects or interaction effects.
Okay, so there you have it. Factorial designs are experimental setups used to study effects of two or more independent variables simultaneously on the same dependent variable. So that example there, I like the visual aspect of the example because really factorial designs are a way to look at a lot of different variables and how they all impact each other within one research design. So other types of research designs might look just at men at low doses of that medication used in the example we just watched. That would just be one research design. But if we want to do something really complex, which behavioral sciences so often does, factorial design is often the way to go. So you can look at how multiple independent variables are all impacting the same thing. This is the type of research I primarily do as a psychologist. We are very rarely looking at just one independent variable on one dependent variable. I'm usually looking at one thing and having several different ideas of what might be impacting it, which is factorial design. So the 2 major types of factorial design, she was talking about incomplete. We're not even gonna go there. The 2 types we're going to cover are full factorial designs, and this is where every combination of all of the levels is tested. So in the example she just used, that would be if she was looking at low, medium, and high dosage of migraine medication for men and for women, and then also looking at diet within each of those groups, that would be a full factorial design. A fractional factorial design is only using some of those combinations. So it's, we can call it incomplete. It does get more complicated, but either way, full is looking at all the possible variables of interest and their relationship with each other, and fractional is just going to look at specific ones. Realistically, most of the time, we design research trying to become full factorial designs, but we don't get enough participants to fit all of the criterion. Like, so for example, her little diagram of, she's saying 2 sexes and 3 different design groups with low, medium, and high pills. If I don't get enough participants in one of those 6 categories, then I can't include them in the statistics, which would make it a factorial factional design, which very often happens in something like behavioral sciences. So some of the advantages is it allows multiple hypotheses at once. So my dissertation right now is a factorial design. I have something like 35 research questions within it. It's stupid, honestly. But it's nice because it saves time, it saves resources. It allows us to look at a lot of different things at once within one participant sample. And it can be a lot more comprehensive. This is really important in behavioral sciences where interactions are so complete. Humans are so messy. So many things impact us. And so being able to have more comprehensive design to account for several independent variables at once on the dependent variable of interest is really helpful for understanding those more complex behaviors. The disadvantage is the complexity. The statistical analysis is way harder for something like this, especially when you're looking at 3 or more factors. It gets massive very, very quickly. The more variables of interest, everything multiplies by that, and it can get very large and be very complex. So there's definitely a lot of statistical know-how that is required to do this type of work. And it requires a lot more people. Like I said earlier, you need to have a really big sample size. Most of the time, you need at least 100 people per subgroup of interest. So going back to that example of low, medium, and high migraine medication for men and women, that's 6 groups. I would need at least 100 people in each of them. So I need at least 600 people minimum, and they have to fall within that range. So realistically, I probably even need more than 600 to make sure I get my minimum in each area. Now, let's say I'm also looking at diet within each of those 6. Well, now it's 12 if I'm saying good diet, bad diet. God forbid I say high, medium, low diet, and now it's times 3. And so you see how it gets very complicated statistically, but also you need a lot more people to do that type of design. And it's very applicable to behavioral sciences, like we talked about. An example here is in a study examining effects of therapy types, cognitive versus behavioral, in session timing, if someone comes in the morning or in the afternoon, on patient outcomes and depression treatment. It's going to reveal a lot more, not just about the effectiveness, but also how other types of variables impact effectiveness, like time of day. So not just the type, but also the time. You could also look at something like gender. You could look at something like which therapist someone views. You could look at something like the training of the therapist. You could look at all that stuff at once in a factorial design. It gets more complicated and you need more people to fill out your surveys and participate in research. But if you can make it happen, it can save a lot of time and money. Some of the methodological considerations, when would I use this? If your research objective really has to look at the interactions between a lot of different independent variables, factorial design is usually the way to go in behavioral sciences. It helps you understand the interactions between those variables rather than just one research question. So if you find yourself being like, wow, that's actually a lot of research questions in my design, it's probably factorial for that reason. Resource availability. So when you have a situation where the researcher can do those types of statistics, again, it takes a lot of know-how. People get PhDs to be able to do that sort of stuff, but whatever. So if you have the ability to do it, it can really help in choosing this design. But more often, people don't do factorial research design because they don't have this resource ability. But if you have it, that's a good reason to use it. And if your variables are really, really complex and you're looking at all these different hypothesized interactions, anytime you're really talking about interactions very much, you're probably going to look at some sort of factorial research design. Strategies to enhance validity is you want to control for confounding variables as much as possible. So there's lots of different statistical approaches to that. I'm not going to hold you accountable for it other than knowing that you would want to be very intentional on controlling for confounding variables statistically, where possible. it is ideal if you can do balance and randomization. So maintaining a balanced design where each factor level is equally represented. That's what I'm talking about sample size. In an ideal world, it's something is going to be most valid if all of your subgroups have at least 100 people in them. But that's not always the case. You can get away with less, depending on the statistics that you use. But for it to be the most valid, most reliable, most internally and externally valid and rigorous, you want to make sure that there's an even balance of people in each of those factorial levels. And trying to randomly assign subjects within them. It's very tricky to do that, but ideally that's what you would do. And you would need to have pretty robust statistical analyses. Again, I'm not going to hold you accountable for the names of any of these in particular, but one way to enhance validity of something like this is knowing what you're doing and using some of these higher end statistics. And the last one we'll cover for the day is single subject research designs. We'll start again with a little bit of a video.
The essence of single subject design is really using repeated measurement to understand an individual's normal variability so that we can use our understanding of that variability to determine what the effects of our treatment are. For me, one of the first steps in developing a treatment is understanding what an individual does. And so if I were doing a group treatment study, I would not necessarily be able to understand or see exactly what was happening with each individual patient so that I could make modifications to my treatment. And understand all the details of what's happening in terms of the effects of my treatment. So that's one reason. So for me, it's a natural first step in the progression of developing a treatment. Also, with the disorders that we deal with, it's very hard to get the number of participants that we would need for the gold standard randomized controlled trial, so that using single subject designs works around the possible limiting factor of having enough subjects in a particular area of study. My mentor was Dr. Cynthia Thompson, who was trained by Leah McGraw.
Okay, I'm going to stop there just for the sake of time, but single subject the research designs like she was talking about is when you just look at one person or a very small group, but you look at them really intensely. So this is a type of intensive study of a single individual participant, sometimes maybe two or three, but usually it's literally single. It's called single subject. So for the sake of conversation, I'm going to say single. And that's when you're really looking at that person's experience of an intervention or treatment over time. So like this lady was just talking about, these can be really helpful when it requires a lot of time and effort to really understand phenomenologically what the participant is experiencing. It's not always feasible to have someone fill out 100 surveys a day. It might be better to just deep dive focus on one or two participants, especially if you're at the early stage of designing something. So very often research, actually almost always good research, is in iterative processes. So you would do one type of research design, come up with your conclusion, move to the next question. That's how science evolves. It gets more and more complex building off of this earlier research. Single subject design research tends to be earlier research within a field because it allows for a really deep investigation of one person, but it lets us know what might be important variables when we want to study it with more people. It's also really helpful when getting participants is really hard. So let's say I want to study someone with dissociative identity disorder. I'm gonna be really hard pressed, even get one person to do that. And with something so complex like multiple personality disorder, it's called dissociative identity disorder, but whatever. With multiple personality disorder, I want to really focus on studying that one person so that if I then have a study later on, I know what parts to focus on. Hopefully that makes sense. There are several different types of single subject research design. So the first one is called A-B design. And this is a very simple design involved getting a baseline. So without messing with anything, let's take a bunch of measurements and then you do the treatment and you test it again. So A-B, baseline, treatment, that's it. An example of this is maybe I find someone with multiple personality disorder. I have them fill out a whole bunch of psychological assessments for me at baseline before I do anything to them. And then I put them through treatment for 6 months, and then I have them take it again. That's A-B, baseline, intervention. ABA, reversal design, is when we have that return to baseline after treatment to check for reversibility and control effects. An example of this is with my same DID, dissociative identity disorder patient. Maybe I test their psychological functioning at baseline, I do a treatment, I test them right after treatment, and then I wait a year and I test them again. So I'm testing their new baseline a year later. It's far enough away from treatment that we know it's not an immediate effect of the treatment, and we want to see if it gets reversed or not. Did that treatment outcome last or not? So ABA. And then we have multiple baseline designs. This involves staggering the implementation of treatment across different contexts, times, or subjects to demonstrate that change corresponds to the introduction of the new intervention. That's a lot of words. Another way to say this is maybe I want to test how well something works, but I'm not sure under what conditions it works. I'm going to use my kind of same example. I'm working with someone with multiple identity disorder. I meet them. I do all their baseline testing. Where are they before we do any intervention? And then I give them treatment, and I give them treatment in a normal setting, like within 30 minutes of their house. They come in person to see a therapist once a week for 6 months. And then I test them. And then I wait a year, I test them again, I get their reversal information, but then this time I put them back in treatment, but I do it on telehealth, but they meet 3 times a week. And then I do the same thing. So now I have this new baseline, we give them a new type of treatment, we collect information after that second treatment, after the telehealth, and then we wait a whole other year. And now we see their baseline again. Is it back to where it was before we started? Is it higher? And then we'll do it again. Maybe this time, instead of telehealth, maybe we'll do group therapy. We'll have them go in person and do group therapy once every other week for 6 months. And we'll do it again. That would be a multiple baseline design. An example of it. Obviously, there's lots of other ways that can look, but there you go. Hopefully that helps. Some of the advantages of this is that it's, again, a very detailed observation. So it lets you get a whole lot of information, like a deep level of information, even though it is about one person. And it also allows for more continuous observation, so you can understand behaviors over time a little bit better. We don't have as much unknown if I'm focusing on this one single subject. It's also pretty flexible, so it's very highly adaptable to individual cases. So that's really helpful for things like therapy or education, where being able to tailor an intervention to that individual learner is really important. So it's hard to do science in something like education, because good science, remember the conversation a second ago, it's going to be very consistent. So if I wanted to do some sort of testing in an educational setting, I would want it to be the same teacher delivering the exact same lesson the same way under the same conditions. But that's not life. That's not what happens a lot of the time. So a single subject can help focus more and gives more wiggle room to change things up, because you're not trying to replicate it so exactly. So those can be advantages. The disadvantages, though, is that it's not very generalizable. Because it is just that one single subject, you can't reasonably conclude that everyone's going to have this experiment. Or go back and remind yourself the importance of generalizability. We talked about it a whole lot throughout the semesters. Hopefully this is not news to you, but just one person cannot speak for the whole. Even if I deeply understand my mother, that doesn't mean I understand all mothers. People are different. And that is a very inherent big disadvantage of single subject research. And there can also be some ethical and practical limitations from withdrawing from treatment. So if I'm working with a single subject research design, there's more pressure on them to stay in research, which can be kind of unethical sometimes, if that makes sense. Very applicable to behavioral science, especially this ABA reversal design, where we're testing things like therapy effectiveness over time. Yeah, we talked about that. And lastly, when would you pick this? So criteria for choosing a single subject research design is usually you're going to pick this when it makes sense to focus on one individual, when it makes sense to do one single subject. It's best suited for scenarios where individuality, variability, and response to intervention, it needs to be really closely monitored for some reason. Again, it's usually used in preliminary early research. So before I design a whole research design for 400 people with dissociative identity disorder, I want to really make sure that I understand that experience so I can know what to control for later. So I'd probably start with a single subject and then ramp up to more participants. It can also be really helpful for behavioral interventions, like we talked about. We're usually a good choice for behavioral interventions, especially for earlier stuff, just because it allows for that more flexibility. Ways you can make it more valid is employing really rigorous and frequent data collections to make sure that it's being accurately captured over time. So if I'm doing my research with my dissociative identity disorder single subject, I don't want to just give them a bunch of random cognitive tests. I want to make sure I'm using the best, most valid, well-known assessments possible so that I can be really rigorous in my data collection, which would make it more generalizable than if I was haphazard about it. Replication is also very helpful. So again, this is usually preliminary research and is later replicated later on so that you can start to address those areas of generalizability. And wanting to think through about any ethical considerations that may impact the participants' welfare or maintaining treatment. My dog just burst into the room at that same time. So that's what I have for you today on those other types of research designs. What you need to be doing is thinking through what would fit your research questions best for your proposed research project for the semester. So continue to reach out if you have questions, and thank you. I see you next time.